Soft background music fades in narrator. Welcome to today's lecture on the fascinating topic of membrane biochemistry. In this lecture, we will explore the intricate details of the lipid bilayer and its components, membrane proteins, membrane fluidity, transport mechanisms, cell signaling, and much more. So, let's dive in. Background music fades out. Professor, the cellular membrane is a complex structure that serves as a barrier between the internal and external environment of cells. It consists of a lipid bilayer, composed primarily of phospholipids. Phospholipids have a hydrophilic head and hydrophobic tails, resulting in their unique orientation in the bilayer. This arrangement provides stability and restricts the movement of hydrophilic molecules across the membrane. Furthermore, the lipid bilayer also includes cholesterol molecules, which contribute to the organization and fluidity of the membrane. Cholesterol interacts with phospholipids and affects the packing of hydrophobic tails, thus dictating the membrane's physical properties. Professor, integral membrane proteins are firmly embedded within the lipid bilayer. These proteins possess hydrophobic regions that interact with the lipid tails, anchoring them to the membrane. They play crucial roles in many cellular processes, including transport, enzymatic activity, and cell signaling. Integral membrane proteins can span the entire lipid bilayer, transmembrane proteins, or associate with one side of the membrane, monopass or multipass proteins. Their specific functions are determined by their unique protein structures and the presence of specific domains that interact with other molecules. Professor, unlike integral membrane proteins, peripheral membrane proteins do not penetrate the lipid bilayer. Instead, they associate with the membrane surface through temporary interactions with integral proteins or lipids. These proteins play vital roles in cell structure, signal transduction, and membrane trafficking. Peripheral membrane proteins can be attached to the membrane through electrostatic interactions, lipid modifications, or interactions with other membrane proteins. Their reversible association enables them to participate in diverse cellular processes. Professor, glycoproteins and membrane glycolipids contribute to the complexity and diversity of the cellular membrane. They contain carbohydrate moieties attached to proteins or lipids, respectively. These sugar modifications are crucial for various functions such as cell recognition, immune responses, and cell communication. Glycoproteins and glycolipids exhibit structural diversity due to variations in sugar types, linkages, and branching patterns. This diversity allows for intricate and specific interactions with other molecules, contributing to the complexity of cellular processes. Professor, maintaining the appropriate fluidity of the cellular membrane is vital for its functioning. Fluidity influences various processes, including membrane protein activity, transport, and fusion. Cells regulate membrane fluidity through modifications such as changes in lipid composition, temperature, and the presence of cholesterol. Unsaturated fatty acids, commonly found in phospholipids, introduce kinks in the lipid tails, thereby increasing membrane fluidity. On the other hand, saturated fatty acids reduce fluidity due to their straight, packed structure. The balance between saturated and unsaturated lipids, as well as cholesterol content, determines the overall fluidity of the membrane. Professor, ion channels are integral membrane proteins that facilitate the selective transport of ions across the lipid bilayer. These channels enable the movement of ions down their electrochemical gradients, maintaining ion concentrations essential for cellular processes such as signaling and nerve conduction. Ion channels possess specific structures that form pores, allowing the passage of specific ions based on size, charge, and selectivity. Different types of ion channels exist, including voltage-gated, ligand-gated, and mechanically gated channels, each playing distinct roles in cellular physiology. Professor, active transport systems actively shuttle molecules across the membrane against their concentration gradients, requiring energy in the form of ATP. These systems are crucial for nutrient uptake, waste elimination, and the maintenance of ion gradients. One example of active transport is the sodium-potassium ATPase, which transports 3 sodium ions out of the cell and 2 potassium ions into the cell, against their respective concentration gradients. This process ensures the correct ionic balance within cells, maintaining cellular homeostasis. Professor, membrane receptors are integral proteins that enable cells to respond to various extracellular signals. These signals can include hormones, neurotransmitters, or growth factors. Upon binding to the receptor, a signaling cascade is initiated, leading to intracellular responses. Different types of membrane receptors exist, including G-protein coupled receptors, GPCRs, receptor tyrosine kinases, and ligand-gated ion channels. Each type of receptor triggers specific intracellular signaling pathways, allowing cells to interpret and respond to external cues. Professor, Membrane fusion is a fundamental process essential for cellular communication, organelle biogenesis, and the transport of molecules within cells. It involves the merging of two membrane bilayers, resulting in the integration of their contents. Vesicular trafficking plays a critical role in membrane fusion, as proteins and lipids are packaged into transport vesicles that butt off from one membrane and fuse with another. These vesicles carry cargo between organelles, ensuring the proper functioning and organization of the cell. Professor, lipid rafts are specialized regions within the cellular membrane that contain high concentrations of cholesterol, sphingolipids, and specific proteins. These structures, often referred to as membrane microdomains, play crucial roles in various cellular processes, including signal transduction and membrane trafficking. Lipid rafts provide platforms for signaling molecules to interact, enabling efficient signaling cascades. They also serve as sorting stations during vesicular trafficking, aiding in the organization and delivery of cargo to its specific destination. Professor, the cellular membrane exhibits asymmetry in its lipid composition, with distinct phospholipids preferentially distributed between the inner and outer leaflets of the bilayer. This asymmetry is maintained by enzymes called lipid flipases, which actively transport phospholipids from one leaflet to another. Lipid flipases ensure the proper distribution of lipids, contributing to membrane stability, vesicular trafficking, and cell signaling processes. Imbalances in lipid distribution can result in cellular dysfunction and diseases such as muscular dystrophy and blood clotting disorders. Professor, membrane remodeling is essential for the formation of complex membrane structures such as invaginations, tubules, and vesicles. This dynamic process requires specific proteins, known as curvature-inducing proteins, to shape the membrane. Curvature-inducing proteins interact with lipids and induce local membrane curvature. They play crucial roles in diverse processes, including endocytosis, exocytosis, and membrane scission during cell division. These proteins ensure the proper morphology and functionality of cellular membranes. Professor, in conclusion, exploring the intricacies of membrane biochemistry gives us a deeper understanding of how cellular membranes function and interact with their surroundings. The lipid bilayer composition and stability, integral and peripheral membrane proteins, membrane fluidity, transport mechanisms, cell signaling, membrane fusion, and many other fascinating topics all contribute to the dynamic nature of cellular membranes. As we continue to unravel the mysteries of membrane biochemistry, we gain valuable insights that can be applied to various fields, including medicine and drug development. So, let's continue our exploration of the captivating world of membrane biochemistry in our future studies and research. Soft background music fades in. Narrator, thank you for joining us today. Please stay tuned for more engaging lectures on biochemistry and its applications. Background music fades out.